So, BioN has been talking a lot about the excellent science that is going on in the region. I think it's time now to shed some light or put the spotlight on some people behind this excellent science, and not just any people. I have Stegle here with me today. I think it's fair to say that you are a celebrity here in the region. You're also a key international figure in your field of science that you're working in, data science and artificial intelligence. And truly, you are also an inspiration to many scientists working in your field. So thank you for having the time and joining us here today.
Thank you for the kind words and for having me. I look forward to the discussion.
Excellent. So for those of you who don't know Oliver, I want to start with a couple of words from your CV. You have a Master of Advanced Mathematics and also a PhD in Physics from the University of Cambridge. You then moved on to do a postdoc for the Max Planck Institute of Intelligent Systems and Developmental Biology in Tübingen. And then from 2012, you sort of dived into the EMBL universe. You became a group leader at the EBI in Cambridge. And in 2018, you moved to EMBL Heidelberg as a group leader in the genome biology. And you are also a division head at the DKFZ and a full professor at the University of Heidelberg. And this is next to various coordinator roles you have. You have gained a lot of very prestigious awards. So I think my first question is glaringly obvious, and the question that is usually asked to women, but I think not many enough men are asked this question: how do you juggle what seems like about 2 to 3 full-time jobs and family? You also have 2 kids.
Oh yeah, it's glad you're asking this. I mean, you know, and I probably give the same answers that most people who ask this question. You're trying to keep the balls in the air and probably always, you know, never being happy with how well you do and giving, you know, justice to any one of your roles. But maybe I think what I should clarify, even though, you know, between EMBL and DKFZ, my lab is 2 affiliation, but it's really 1 lab. So I think I see myself, we have 1 group that is trying to bridge and bring together and leverage the excellence of 2 institutions, which are very complementary. So I hope it's at least 1.5 jobs and not quite 2. But yeah, it's sometimes difficult if you don't come home and your kids ask you why you're away again. But you know, I guess
that's familiar.
Yeah, that part is not solvable. Yeah.
Yeah, it just depends on how you put your hats, I guess. Okay, so now looking at your CV, mathematics, physics, kind of frightening for a biologist at first, but then luckily you very quickly also dove into the life sciences, where it's a field that is familiar to me and I guess to our participants as well. So today your field of work, the research that you do, you're interested in putting some order into, you know, the mysteries of genetic variations. You're interested in how these variations develop and look at them over time and generations. You also look at variations between species, but also in an individual. These, you know, variations can occur, for example, in the context of disease. So can you tell us a bit more on how your passion for numbers changed or developed into a passion for the biological system?
Yeah, I mean, it's a very personal story. I mean, I was, as you mentioned, in Cambridge in an applied maths program working on quantum information theory, which is basically, you know, developing error correcting codes for quantum computers. And at that time, I thought there will never be a quantum computer in my lifetime. I was wrong, admittedly, but it pushed me towards a field where I could see, you know, we can have impact today and now. And I was fascinated by biology just as this sort of messy discipline with all its facets and complexities. It's very different to physics in many ways, but he's still trying to bring on order. And I think that's what you're doing as a physicist, right? You're trying to find out what are the basic equations of life, if you wish. I think I've given up on that quest to find basic equations, but still I think it motivates to study variation. It motivates to study what causes differences, how can we make sense of these differences. And I think it also motivates one of our passions, which we actually, you know, I started in Tübingen in my postdoc, and now I come back to, which is actually to try and understand causation. You know, what causes what, and I think that's something where you know, physics has basic laws of life in a way, if you wish, they govern that, but in biology, we can also now draw these inferences and really understand how genetics impinges on cell states, how genetics increase disease risk, or how genetics interacts with environment to then up or down regulate our risk for getting diseases. So these questions, I think, are quite exciting, and I think they still leave some sort of tangential rooting and some physics-inspired principles, although I've traveled quite far in physics.
So now we're entering the field of artificial intelligence, and artificial intelligence has been around for many years, I think from 1950s on. I think you could probably mark the term artificial intelligence, but it's usually known in society or associated with, navigation systems, self-driving cars, ChatGPT, and so forth. And it has only gained significance for the biologies, for the life science recently. And then it has really changed the world of life sciences and how you coordinate data and so forth. Can you tell us a bit more about this? How has it changed your work? How that will impact on research?
Yeah, no, I mean, very, very happy to. I mean, maybe I should first say that in a way, what we have been doing, you would always, from today's perspective, classify as AI and machine learning. Although that was in the infancies of that field when it wasn't such a big thing, and really was about modeling dependencies and probabilistic modeling. And I think what has now changed is that the assumptions we're making about biology, they can be weaker and weaker and weaker. And one of the reasons why we don't have to make such strong assumptions is data, the explosion of data sets, which I will come back to in a minute. And the other one is we just have made technological improvements. We have now different ways to model and to derive these inferences, which gives rise, I think, really to the success of AI. And we have the hardware to actually do the implied computations and to really train these models at scale and otherwise. But I think really the driver of this change is data at scale. I mean, we see in biodata doubling of data volumes every 18 months or so. I mean, that's at least the numbers from the EMBL archives in Kingston. And that really happens across all domains, from structures to molecules to genomes, and these increased volumes of data have been able to tackle questions very differently and in a data-driven way. What's also, I think, more important to say is that we have had a massive community effort over decades to think about what are good questions and what's right and what's wrong. And what are, we call this labels, the ground truth in which you would want to predict. And one example is, for example, I think that the breakthrough in AlphaFold is not my field, but I think it's still, probably the example you have to cite when you talk about AI and biology, predict structure, protein structure from amino acid sequence. And I think it's such a great example because it shows how a decade-long activity to collect protein structures and their sequence, how that activity, including benchmarking and understanding what are structures and what are the corresponding pairs of sequence and structure, how this actually has then fueled the success of AI. And we're seeing this now in many other fields. And my domain is genetics and single-cell biology, where we have similar such problems and questions, how we predict from the genome what happens in the individual, what happens in the cell. And these types of questions can, I think, now increasingly be tackled using AI as a tool. And yeah, I think we have to embrace that. It's a new world, and it would be foolish to not embrace those tools.
Yeah, it's a bit scary sometimes. But concretely, so for our listeners, what does this mean for, you said you can look at variations. So that helps then to see what happens in disease, for example. And so where's this going? How can this really help in an applied way for patients, for example? What difference does it make?
Yeah, I mean, I give an example from our own work. I mean, the traditional field of genetics, right, is, where we started, now it's 10 years, the lab just turned 10, which is quite a scary time scale. We compared essentially individuals that carry a particular variant and those who don't carry. And you can ask them the question, is for those individuals who do have a particular feature in the genome, one, we call them single base pair change, where one of your 4 letters, it differs, whether those individuals that have a particular genetic feature have a higher or lower risk to develop a disease, for example, Parkinson's disease later in life, or something benign like ERX type or BMI. And many of these things have a strong genetic basis. Up to 30% of our weight is heritable, which is, I think, quite important to know and also a bit scary in some extent. But what's now the real challenge with these problems, with these types of approaches, is you need two big groups. You can only make comparisons if you have a decent number of individuals that have this variant and that don't have it. And that just limits what you can look at. The genome is a big place. It's 3 billion base pairs. Most variants we have are private or really, really rare. You find a variant, you have once in a cohort. You can biobank has 500,000 people. Sounds big. There's still, most variants are private or rare. One individual, 2, 3, 4. And you can't do the comparison. You can't say, oh, well, let's compare these 4 individuals to my 499,996. This doesn't work. And so you need methods that basically go beyond being able to compare two groups, that can de novo from the sequence understand what does a variant do I've never, ever seen before. And this is really where AI comes in. We can make, for the first time, meaningful and increasingly accurate de novo inferences about pieces of the genome you've never trained these models on, about variants you have never, ever seen before. And that's really exciting because you can go into this sort of, I call it personalized genomics. You have one genome from this patient, and you can't compare this patient to anything meaningfully. You just want to see, oh yeah, there's these variants, and which of these needles in the haystack is responsible. And their AI is a great tool, to find things that could be the cause. I think it's also important to say it still could be. It's predictions. Not all predictions are true, but it's a great place to start compared to looking at this vast number of unordered hypotheses. I hope this was an example too.
No, I think very well explained. So this is diagnostics. So this is maybe not your field, but also, AI can play a role in treating diseases. So making predictions as to what drug or what dose of a drug could be efficient for a patient. So maybe some words on that.
Yeah, in a way, the fundamental problem is somewhat related, in that you would like to sort of solve these counterfactuals. What would happen if we want a what happens if question and answer for that. And here the question is, what would happen to an individual with the specific genetic background, maybe the specific lifetime coverage, maybe the specific health records, and the intervention is, in this case, the application of a drug. And that's actually something we also to some extent tackle, by integrating these different omics modalities, something we've been doing for a long time now, and try to find answers to these sort of counterfactuals. And it's a similar problem though otherwise, that you want to really go beyond what you've seen before. You don't want to say, what's the most similar patient I have, and just look what happens individual. You want to answer a question to intervention you maybe have never done before. And this is where AI can extrapolate beyond what we could previously do. But there's obviously important caveats, right, to be also aware of. These methods are imperfect. They frequently don't know what they know and what they don't know. And that's really important. We call this calibration.
Yeah.
You want a model not only give an answer, but that you can deduce how accurate is that answer. And this is a big challenge, and I think we're just starting, and also from a very theoretical point of view, that to get these models to the level of robustness that we can really use them in the clinics. But there's great promise.
Yeah, especially for diseases where you said you don't have the numbers in patients and rare diseases, and where there's not much treatment hope otherwise. So, that sounds really exciting outlook, also for applications in medicine. Many people talk about the dangers of AI and where this could go. Do you have an opinion on that? And are there maybe efforts that we can do to constrict these dangers?
Yeah, it's obviously a question that's on everybody's mind. I think it's also really important to maybe classify for one moment a little bit what different AI does.
Sure.
The type of AI I was just describing in a way is if you wish, we call this a classification problem. You want to hunt the needle in the haystack for once, or you want to make predictions about what would happen in an individual. And this is in a way a tool. I think one has to be sort of strictly, confining that there's a difference whether we use AI to create hypotheses and understand which hypotheses are more likely to be relevant, versus if you give a machine autonomy and autonomous decision power. And that's, for example, in driving a different world, it just makes a difference whether a clinician gets recommendations or whether you put an algorithm in a car. And I think that's one differentiation to make. But even if we just make recommendations, we need to be really sure that these recommendations satisfy quality metrics. I mentioned calibration. You want to know what you know and what you don't know. It's not only accuracy, but also elements of, for example, implicit bias. That is important to understand. If we train algorithms on Central European populations, they may not work as well in other populations on this globe. And we need to understand that and make them as fair as possible. And I think another dimension that also really is linked to that is, at the end of the day, is to understand the black box. I think in a medical context, we have I think, very rightful natural intuition that we want to be able to unpack how does this machine come about making that recommendation. And the better you understand that scientifically, and even ultimately maybe explain it to the patient you're treating, the more confident we can be and can use these approaches. And these are things we have to do well. And we have to also develop standards. We have to develop best practices. And at the end of the day, there are also ethical implications, as I mentioned. What do you do in other populations? Do you use a classifier that you know right now can benefit some individuals, but it's maybe only some Central White Europeans and others may completely not benefit? Is that now sound and acceptable to deploy that already in the clinics? How do you make sure you close the gaps? And these are many regulatory questions that are also not new in medicine. We had, for example, now an important act on a European level that pharma has to prove that new drug targets are not applicable in children. And that's exactly the same reason you want to avoid this bias, that medication is developed where the returns are largest and others are left out. And I think AI will have a lot of these types of problems. That it works in some individuals, and we need to make sure that the regulatory systems are set up in such a way that we mitigate these biases as quickly as possible, without sort of closing the door to massive breakthroughs and that we can otherwise utilize and leverage.
So then you're talking about the EU Artificial Intelligence Act. So that's a good thing. Like, are you feeling that's doing what it's supposed to be doing? It's not, restricting your work. What's your opinion?
I mean, it's a very ongoing discussion in the community. I mean, actually, I was referring to an act which I can't put my name on, the finger on his name on, that does about molecular targets and drug discovery at the EMA level. The AI Act is, I think it's still a very fluid in terms of what are the implications it really has in practice. Some regulation will be important. The obvious challenge and question is how much freedom to preserve, when to regulate, and how can you make sure that innovation is just not stopped? I mean, if innovation leaves Europe and is done elsewhere, we have not really gained anything, because then we are participating AI as passengers in a car that travels, and we're really no longer on the driving seat. And I think that's what many people are worried about, that the hurdles are so high that we essentially don't participate anymore in the innovation, and that would be very sad. And I think there's a lot about detail how that's implemented in practice and how that is also put in national law. We've seen similar like data protection.
There's other fields.
The devil is in the detail, and that detail is very important. And I very much hope that there's going to be 16 different implementations of the AI Act in every state in Germany, which is what we've seen in data protection and caused a lot of grief.
Yes, and we're just about to fix it hopefully soon. Okay, but so this is about your research. Thank you so much for the insights in AI. I want to go back to your early career. It seems like a bit of a love-love relationship between Germany and UK. You've been changing, around, to Cambridge, back to Germany, back to Cambridge, back to Germany. You've then probably observed a lot of differences in the different systems. How was that?
An interesting ride. I mean, I think it's the same experience that anybody has made who's lived substantial time abroad. You look differently at your home country and you wonder, why don't we do it like this? And then you still live in a country where we desperately miss good bread and pretzels. And I always wonder why we don't export scones from the UK to Germany and get the good pretzels into Britain. But anyway, I mean, there's many of these benign questions, but they obviously carry through from daily life all the way into scientific systems. And you have exactly the same questions. You wonder, why can't we just mix and match and get the best of both worlds? but these things are equally hard in science than they are in bakery. I think that's I think the reality one has also to accept, and both of these worlds have their pros and their cons. I at the time took a personal decision. I was very much at home in the British system, and I very much enjoy and appreciate certain elements in the German system. So yes, I think it's useful in the way because you can bring in some ideas into another system and incorporate. And I think Heidelberg is a great place, both with institution like the DKFZ, which is really a national champion and actually a European champion in cancer research, and has a lot of also possibilities to impact and influence elements. Like we, for example, coordinated data infrastructure, making data available and shareable. That's something that really was missing from a British perspective. And then EMBL up the hill, which is a European organization. And I think bringing these worlds together, a European perspective into Germany and then back, I think that's what I took away. What I tried to also contribute a little bit, that we are don't lose this outside perspective. Sometimes you get very much inward looking into your own country, but that happens in the UK as well. So
yeah. Any specific examples where you think what Germany could learn from the UK and vice versa?
Yes, well, up to a certain day when which was I think Brexit is a bit the elephant in the room. Up to that day, I really felt Germany should just learn to be pragmatic. I think there was a counter example that also Britain can be non-pragmatic in some way. But I think in science, pragmatism and finding pragmatic solutions is a very, very strong driver. And that has been something one could embrace, I think, still more, particularly when it comes to things like recognition of degrees. I think when we spoke about this interview, I briefly mentioned, my first degree ever was actually a PhD. Because in the UK, I was admitted to a PhD program without any degree. And that would be unthinkable in Germany. Absolutely unthinkable.
God forbid, yeah.
And these sort of elements, I mean, they're being pragmatic and recognizing talent and also diversity, what talent is, and that, they're different people, and it's good that there are differences. This embracing diversity in a positive way, that would be something I really wish we could do, and it would be easier to live that, because we all try to bring it to the system, but then there's a rule, and it's sometimes difficult to cross certain lines where everybody agrees we should be able to cross it, but it just doesn't happen.
Yeah, I think Germany is still world champion in regulation, so something we can maybe look into. But you're here now. You decided to stay in Germany. You are in the region Rhein-Neckar. You've already mentioned the DKFZ, EMBL. So obviously our region is very special in many ways. There's a high density in academic science. There's also industry very present. Have you taken advantage of that? How do you think is that impacting your work to have those two sides, academia and industry?
Yeah, it's a good question. I mean, I have to say, I'm really a basic scientist, and I really develop computational tools that I think are useful equally in industry and in academia. I'm regularly invited to industry to present on our methods and give advice on them, and sometimes we have also collaborations around those. So for me, you can really see that there's now a much more sort of equal playing field in terms of the approaches, how industry and academia is run. There are some differences, but there are also, I think, an impressive degree of convergence. We have a project with, for example, GSK here in Heidelberg, and it's really a collaboration like you would collaborate in academia. And I think that's a very positive element, and particularly if you see that some of your science maybe can be taken up and translated faster, more efficiently. So I really appreciate that part of the ecosystem here, and just as mentioned, the density of just bright people who want to make a difference in vastly different fields and areas. What I think we could do even more so is having sort of more structured interactions and activities. I think it's great that BioRN exists and that you're trying to facilitate as many of these connections as possible, but I think the UK is still a bit more dynamic at this point in time. That's fair to say, and that would be great to learn.
Golden Triangle is definitely a region that we look to and try and get the most of here as well. Okay. So you mentioned interactions with industry, so that's public-private partnerships. The GSK Corporation, is that open targets? So
No, it's a different
example. Yeah.
It's a separate collaboration, really on a project level, in part of a framework agreement between GSK and EMBL. But yeah, exactly. I think something like an open target in Heidelberg would be on a different question.
Okay.
And I think, for example, AI is a really interesting area also for pre-competitive funding, because at least for companies whose business model isn't the AI itself, like DeepMind or others, I could well imagine that this is a topic one could consider as something we tackle collaboratively in a pre-competitive state, because everybody needs these tools. You need also maybe influence where academic innovation happens, but then use these tools to then drive the competitive side, which is then happening in a different part of the product development cycle. So I think that could be really an interesting dimension, and I think there maybe one day we can.
We'll take a note.
Yeah.
Maybe something that BioM can facilitate in the future as well. So in many other conversations, sort of one theme has come up in Germany is the bottleneck really for research and applied research is the lack of funding. Is that something, so I mean, you work at EMBL, EMBL is well funded, I assume. Is still the lack of funding, especially in translational research, something that you have observed? Maybe you have found, interesting solutions in other countries that you've met?
Yeah, I think it's first, I think important also to say a little bit, that I'm a computational scientist, so we work with not quite with pen and paper, but develop algorithms and develop code. Our main cost factors, our main bottleneck is people, and followed by compute. And so this is a very different regime to translational research that, for example, requires large patient cohorts, where there's larger money in other dimensions. I think we are somewhat less affected by perhaps the lack of funding. And the thing when talks lack of funding, it's also very important to really specify that a bit closer. My experience from UK versus Germany was that individual level funding for one project, one student, one postdoc, actually it's much easier in Germany. And Germany is extremely wealthy. What is much harder in Germany is to get strategic funding for one topic, to nucleate a new institute, to nucleate a large program, to get something done in a period of 5 to 10 years. This is where we are very, very, I think, slow, and also the volumes are lower, particularly also industries involved. So I think it really depends. Funding my PhD students is actually much easier in Germany.
Basic science is usually not such a big problem, yes, in Germany.
Yeah. Or expensive basic science is also a problem. Yeah. I think it's basically large grants, large sums of money that enable things, that require substantial investment, a strategic investment. That's I feel is something where the UK has more players who support that type of activity.
Where it's coming a lot from private resources, right? So that might be a good source of money that maybe in Germany we can mobilize a bit better. You mentioned the funding of research institutes. They seem to be popping up in the UK. Every year there's a new research institute. I think 2 years ago, the ARC Institute. And now just in January, there was an article I read about someone from the Broad Institute now opening its own with a private sponsor, a research institute. So is that the way to go in Germany, to have these privately, independently run research institutes?
I think every country now needs its own system, and also solutions to specific problems. And there's I think a very important difference that's just enshrined in our current cultural understanding, and that's basically how to deal with people and personnel laws. The level of tenure you get in a country like Germany, and frankly that even also includes industries, is very different to the UK or the US. Actually, many countries around us, even Austria, Switzerland, have a very different understanding. And that makes it harder to open things and close them again. And in the UK, you see this closing actually not only for private money institutes, but even the MRC or the BBSRC, the public funders, equivalent to the DFG. They scrutinize their institutions quite heavily, and they shut them down. And this is something which is comparatively rare in Germany. And obviously, you have to be able to shut things down to open things again. And I think that's there's many reasons why this is structurally harder. I would imagine that in Germany, unless one wants to change these basic parameters, one could imagine more, sort of hubs, actually maybe more what also the broad has been doing, where people can be seconded to new things. And the secondment might not have the same bearing on labor law. And that means that you can maybe shut this thing down again in a couple of years. So I think we need to find solutions. And I think there might be different solutions, but I definitely agree we need more agility in creating things, and then also being willing again to make them disappear without causing grief to personal lives and these other issues. I think that's definitely a good point. Yeah.
Yeah, and maybe also to use the critical mass that we have here better, combining things, and that's where the Health and Life Science Alliance here in Heidelberg Mannheim is going to, and you're also a coordinator. You're playing a role in that as well. So maybe some words on that, and then we're handing over to the audience to ask some questions in the chat. I think we have already some, but maybe let's give that couple of words on that.
Yeah, I think this alliance is really just maybe a few words for those who have not come across it. It's a really a strategic investment and effort, very much supported by the Land of Baden-Württemberg, the region, motivated also by the merger of two, to create an ecosystem that not only has the translational domain in terms of patient care, but then also the non-university institutions under one framework and umbrella, to mobilize synergies across institutions. You mentioned the high densities of partners. If you put Heidelberg and Mannheim together, it's really quite staggering what density is here. And this effort, the alliance, is unique in that it also resembles really a legal entity and tries to basically bring together currently disconnected pieces, I think, particularly between the life sciences and the medical disciplines, the computation between medical institutions and basic science institutions, and foster this interdisciplinary bridges. And I think it's a great experiment. I mean, it's early days. We just started. I was coordinating the AI arm of the alliance in the early days, and now it's all coming together on this new roof and this new umbrella. And I think we have seen great successes in creating new projects, getting people to work together, recruiting fellows to Heidelberg, creating even new staff categories. And we had this one branch which we called AI clinician scientists. So clinically working scientists who then also want to use their expertise based on AI, bring AI to the clinics. And so I think these are really experiments we cannot do in this framework. And now it's the time to go from experimentation to also sustainability and create a sustained structure. And I think it's an exciting time. And I really hope that some of these elements we talked about could be realized in this framework.
Through the alliance.
Through the alliance, yeah. And create this as a hub in the middle that can tackle ambitious questions, that can be temporarily home to scientists, who then, but also after some time, also go back to their day job, quote unquote. I think this is one of the many directions this could go, and I think it's exciting.
It has great potential, especially when we take industry into the circle as well.
Yeah.
Okay, I am reading some questions from the chat. Thank you, Oliver, for this great interview. A long question is coming. I'm wondering your opinion on synthetic biological intelligence. These are essential systems that integrate biology, neurons into machine learning. Two recent published examples of this concept are neurons that were taught to play a simple video game and to a neuronal organoid that was trained to distinguish between different spoken voices.
Yeah, I mean, I want to pick up two elements from this question, which I think is very interesting and thought-provoking. One is really synthetic. I mean, maybe I just want to just start very broadly. I think synthesizing biology is, I think, one of our great frontiers ahead. Research variation, you mentioned it. I've been doing this for now a decade, and I think what we now have to do is to synthesize and create biology at all levels. We synthesize genomes and genomic variation. And the example of this question was mentioning about synthesizing neuronal connections to basically rewire neurons in different ways to create intelligent systems. And I think we need to embrace this in the fullest extent. And it could really help us to even solve societal challenges. Maybe video playing is not one of them, but we can synthesize organisms to convert CO2 in a faster way. So I think synthesizing biological system is a basic science effort. And I think even at the moment, these toy light examples are great to shed light on that. I still think we will do probably compute on computers, although I know there's some promise that these biological neural networks could be more energy efficient. So I think it's a great thing to engineer life, and let's see what.
There's a new institute in Heidelberg, Engineering Molecular Systems, that is picking exactly that up. So again, Heidelberg at the forefront. Nice. You wanted to pick it up on two different levels. You said that's it. Okay, excellent. So we have the next question, which is again 2 questions. How could AI contribute to predicting the function of the 98% non-coding of our genome sequence? And second, how do you want to use deep learning in rare mutation to predict a new disease? I'm not sure I understand it correctly.
Okay, well, that's a very, very deep question, specific question. I mean, happy also to follow up on that offline. Yes. I think just to give a glimpse of that, what we can now do. I mean, historically, the coding genomes, these are these, regions in the genome that are around our 20,000 or odd genes that basically code for proteins and cause function in the cell. The non-coding genomes for listeners who are not familiar with this is basically the space of the genome that's not directly coding, which is the vast majority, but could still impinge on regulatory processes and change the way how these other regions, the other 2%, function and how genes are expressed. And what the AI is already doing today and how we're using it, but really the whole community, is that we have now models that get directly from sequence, and that sequence can be a piece of coding DNA, but actually AI is much more important even in the non-coding space. Directly from the sequence predict a molecular function, for example, whether an enhancer is active, whether a transcription factor can bind, whether splicing happens. And these models that we can now train from our observational data, from observing and surveying molecular biology in large data sets, they can be used to really ask questions about genetic changes everywhere in the genome, including the non-coding space. And this is how we can make predictions, and this is how we can try to get a handle on where these variants are. I hope that gave you a glimpse of what's going on.
I think so. Do we have one final question? Okay, let me see. You mentioned causal models. Where do you see the current opportunities and limitations when it comes to applying causal models to biological data? Where does it work and why? And where doesn't it work?
Yeah, great question. Again, that's a small afternoon conversation we should have here. I just want to give you a glimpse. I mean, I started this during my postdoc, and obviously causation is this fundamental question. We know the 2 variables are correlated. We want to know, does A cause B or B cause A? And there are now really new opportunities in biology to use causation, because causation is important, because we can only intervene, for example, in the context of developing drugs, if we know what is the causal player and what is upstream, what's downstream. For example, many molecular changes occur in response to disease, and these are really bad targets for a drug because you don't fix the problem. You need to find out what is the cause, what drives these things. I think the key opportunities are twofold. One is genetics. I think many companies in pharma around the world using this open targets, as mentioned before, it has heavily been motivated by genetics. Our germline genome, the genome we inherit at birth, we call it an instrument because it's immutable. It doesn't change. Our genome doesn't change in response to disease, with the exception of cancer. And that gives you a very nice angle to do causation, and it's a very, very powerful paradigm that motivated, as we just discussed, a massive, I think, public-private partnership to mine genetics. I think there's a new world, and that is engineering. We can now engineer systems. We can knock out genes using CRISPR. We can change genomes using CRISPR. And that is the same thing, that we can really reverse the causation. We have the handle and changing a bit in the cell and seeing what happens. And this is really where causal inference works. And this we use this a lot now in driving causation, and I think that's a fantastic area. Where it doesn't work is almost everywhere. It's just a hard problem. I mean, causation is extremely difficult, and we have to make progress. And I think we need instruments. We need the ability to manipulate a system to really make causation work. Okay. Very briefly.
Well, fantastic. I think, thank you so much for shedding some light on AI and making it slightly less scary for the biologists out there. I think we're wrapping up here. There are no more questions in the chat. As we said, there is the opportunity to download this video from our webpage later on. There's also opportunity to get in touch with you if there's other questions, and we're happy to pass them on to Oliver. Thank you so much for your time,
Yeah.
for the insights. It was a pleasure.
Thank you for this interview and discussion. I really enjoyed it, and thank you for the audience and asking thoughtful, very deep questions. And I look forward to maybe meeting some of you.
Great, and I see you guys back here, or my colleagues will. The next M2M from BioIN is on the 6th of February and featuring Value Data, one of our BioIN members. Thank you so much for being here, and see you soon.